All right. So I'm going to talk about the pan-cancer analysis of whole genomes project. I am having some electronic issues here, which I will fix. All right. Okay, so I fear we may be a little bit cut off here on the right side, and I can't do anything about that. I'm going to talk about the PCAWG, or Pan-Cancer Analysis of Whole Genomes project, which was a large consortium project involving over 700 researchers over 5 years, in which we took the first really deep dive into the patterns of somatic variation and germline variation in the entire cancer genome, totaling over 2,600 whole cancer genomes across 38 tumor types. We generated 800 terabytes of sequencing data, all of which has been released to the world, and published our results in last February 2020 in 6 Nature papers and 16 companion papers that appeared in various Nature Publishing Group journals. So the reason that this is an important project is because, as I think everyone here knows, cancer is fundamentally a disease of the genome. It actually starts out at birth with inherited germline variants, some of which will increase the risk of developing cancer by damaging the DNA repair mechanism. Over a period of time, both in people with germline risk and just normal people on the street, somatic variations arise in every cell of your body at a low but steady rate. Most of these mutations are passenger mutations. They're not functionally significant, but every so often you get a driver mutation which hits a gene which is responsible for cell growth and regulation. This causes a clonal precancer proliferation of cells, and over time, additional mutations, driver mutations accrue until you hit a critical point at which you start to see rapid growth. And at that point, the tumor becomes visible radiologically and clinically. By the time the tumor has reached clinical attention, most tumors are multiclonal. They have different combinations of driver and passenger genes, and this then begins a process of selection and evolution. You can treat the cancer at this point and kill most of the clones, is not susceptible to treatment. This then proliferates, and the cycle continues with additional mutations, selection, and tumor evolution, and is expressed as recurrences and distant metastases. So we've been sequencing genomes for the past decade, primarily the coding portion of the genome. And over the course of the time, we identified, as a community, roughly 600 cancer driver genes. P53 and KRAS and adenosis, polyposis coli, and so on. But the work has only really covered roughly 1% of the whole genome. Pretty much all of what we know about cancer genomes is in this little teeny tiny sliver of the genome, and all the rest, which contains important functional elements, including non-coding RNA genes, enhancers, telomeres, various regulatory sequences, and then the kind of the dark matter of the genome, which are transposons and defective viral sequences. We really haven't studied these in the context of cancer at all. So the Pan-Cancer Analysis of Whole Genome project was established to answer a slew of questions about the dark matter the 99% of the genome that's non-coding, and its role in development and progression of cancer. The primary questions we wanted to ask are, how do non-coding elements contribute to cancer? What are the causes, effects of structural variations, where a piece of chromosome becomes rearranges with another chunk of chromosome? How do tumors get initiated and evolve from their cells of origin? What are the signatures of mutations, and how do they do variations at the somatic and germline levels influence patterns of mutation and DNA repair? We were really interested in knowing what effects do variations in dysregulatory elements have on the expression of nearby genes and their can we explain by the combination of coding and non-coding variation. Now, there's enough here for 5 or 6 talks, so I'm going to focus on the non-coding that can be explained by coding and non-coding variation, and then I'll give you some practical examples of how this information can be used to improve patient care. The prep work that we needed to do to make PCAWG possible. So we started out by marshaling all the whole genomes that had been sequenced by the combination of the International Cancer Genome Consortium, ICGC, and the tumor and the Cancer Genome Project, TCGA. Ultimately, we pulled together 2,800 whole genomes, and that's a tumor normal pair, so it's actually 5,600 total. And about half of the donors had whole transcriptome data accompanying their whole genome data as well. We uniformly reanalyzed each read set from the very beginning, from the alignment stage to the variant calling stage, and I'll explain to you why we needed to do that in just a second. Because this is a large number of genomes and a large amount of data, we developed a cloud infrastructure so that we could distribute the computational load across various sites on the globe, and also did the difficult but necessary work of harmonizing the histopathology descriptions of all tumors, so that a luminal A breast cancer from one group was the same as the luminal A breast cancer from another group, and so on. And then organized teams of 700 researchers into a set of working groups focusing on different aspects of tumor biology, which was a task in and of itself, and then coordinated a series of publications using a process of internal peer review, looking at manuscripts that were overlapping in research area, and doing some mixing and matching and matchmaking, and quite extensive negotiations with editors to come up with a final set of papers. So we started out with 2,834 cancer genomes. They were from 48 different sequencing projects around the world. This shows you the distribution. There were 38 unique cancer types altogether. An important thing to realize is that some tumor types are more common than others. So we had lots and lots of breast and lung cancers and prostate cancers, but very small cohorts for some of the rarer tumors, such as the sarcomas. So it turns out that harmonizing variant calls in whole genome sequencing data is much more challenging than doing the same thing in whole exome sequencing. And the challenges are, first of all, the non-coding genome is highly repetitive, lots of transposons and line and sign elements, which screw up alignment and mapping. Also, because of the costs, the coverage of the genome is low for whole genomes. It's typically 30 to 50 fold versus 100 to 300 fold in a typical exome project. There are regions of high GC content, which which have lower coverage. And when you look to see how to do the variant calling, and you go through each published method for somatic variant calling, each published method which has really good benchmarking data that shows that it is the most accurate one of them all. Clearly, they can't all be the best. And so we started out by just looking at the you may or may not see the x-axis here. My screen is slightly column is a different variant caller. And we're showing the number of single nucleotide variants or SNVs called by each one. And they're color coded. So those that were called by 10 out of 10 mutation callers are colored purple. 9 out of 10 are colored dark blue. 8 out of 10s and all the way down to unique ones, which were called by only a single mutation caller and not by more than one. And first of all, you'll see huge differences in the number of SNVs. Of consensus calls, and that some of the mutation callers are very conservative, while others are highly non-conservative and call a lot of singletons that are not seen by others and are probably false positives, but we don't know for sure. So how do we deal with this? Well, we dealt with this by sampling each of the classes of SNVs, the high consensus all the way through the singleton ones, creating a hybrid selection chip, and then doing experimental validation on the individual calls, so that we had a gold standard set of validated mutation calls, sampled from the whole set. And then when you put them up on a precision sensitivity curve, you see large differences in the precision and sensitivity. Some callers are doing quite well up here in the upper right-hand corner. Others are trading off low sensitivity for high precision and vice versa. If you look over at the indel, these are simple substitutions. Here are indels, and you see that there's a wider spread. Furthermore, none of the mutation callers is doing much better than an F1 of 0.6 in this case. So what we did then is we selected the best variant callers from each set. We selected these three: DKFZ, Sanger, UTEC, Sanger, and Washington University, Wustl for here, and another set from for indels. And then we attempted to combine them as ensembles in order to improve the accuracy. And so this illustrates for indels the raw accuracy of three of the callers: DKFZ, Sanger, and Smuffin from Washington University. And then various ways of combining them. You can do the union, which increases the sensitivity but decreases the specificity. You can do the intersection, which does the opposite. And then you can do various types of machine learning on these decision trees, random forests, SVMs. And we identified different methods for the SNV and for the for indels to combine the ensembles into a better predictive model. So for indels, it turned out that the SVM gave us the best overall combination. For simple substitutions, it was the algorithm that worked best was the best 2 out of 3. Okay, and then we put this together into a Frankenstein model. in which we started out the BWA-MEM program, did a set of filtering for some of the mutation callers that required it, and then we ran each of the mutation callers on the appropriate subset of tumors. Following this, we did some filtering and cleaning. This step is, for example, is removing oxidative damage artifact. And then finally, we combined and annotated them together to create a set of consensus calls that had relatively high accuracy. Some of this was from data transfer speeds, others were the computation. And so in order to minimize the computational load, we spread it across multiple clouds located in 3 different continents. And it was a combination of academic clouds, such as the Cancer Genome Collaboratory in Toronto, commercial clouds, such as the 7 Bridges Cloud and Amazon Web Services in Virginia, and some traditional high-performance clouds, high-performance clusters, such as the Barcelona Supercomputer Center in Barcelona. And we developed a whole scheduling infrastructure for the collected and distributed the data to downstream analysis via a series of compute clouds. The one I'd like to highlight is a free academic cloud located in Toronto. It's the Cancer Genome Collaboratory, where you can get access to the entire data set, both the raw reads and the interpreted reads and variants. And to get started on that, you would go to cancercollaboratory.org. Okay, so let's talk about what we found in this data set. So the final data set, after filtering out low coverage tumors and tumors that had artifacts, were a total of 2,583 tumors on the white list, 43 million single nucleotide variants, 2 million indels, and 288,000 somatic structural variations. And as previously reported, there's a couple orders of magnitude from one tumor type to another in terms of mutation density, going from hundreds of mutations in pediatric brain tumors up to nearly hundreds of thousands to millions of mutations in high mutational burden tumors such as melanoma and lung cancer. The next step is to distinguish passenger mutations from driver mutations. So there can easily be tens of thousands of mutations in a tumor, and the vast majority of these are passenger mutations that have no impact on the cell. We don't want to study those necessarily. We want to study the driver mutations that are functional and affect cancer-related pathways. Unfortunately, distinguishing them is quite challenging. There have been multiple methods developed for this. One can calculate a background model of the mutation rate and look for tumors that are expressing a higher number of mutations than expected by chance. There are also methods based on looking for variants that have functional consequence. So they they disrupt an enhancer motif, for example. And there are also a few methods which look for unusual clustering of variants in functional elements, such as a bunch of mutations around the splice site. As we did for the variant calling system, we were very deliberate in this, and we did a benchmarking first against a set of known driver mutations, and then we worked out ways of combining the various driver calling methods into a consensus set. And then we went through a manual process of examining, annotating, and filtering each of the putative driver mutations. And we did this both on a pan-cancer cohort where we have all the samples, we have a high N, but a lot of heterogeneity because we're looking at different tumor types, as well as tumor type specific cohorts, such as looking just at liver cancer, where we have a smaller cohort size, but a more uniform biology. Genome, because you'll lose power to multiple hypothesis testing, we looked only at curated functional elements, including coding regions, of course, because it's there, promoters, enhancers, untranslated regions, microRNAs, long non-coding RNAs, and a couple of other functional elements. And we also looked at unbiased genomic segments, where we found hotspots of mutation. And then there was annual inspection and filtering protocol. These are important for removing drivers that are in regions that we know are susceptible to certain localized mutational these were manually inspected and sometimes removed. We ended up running, I think this is about 16 different mutation callers listed here. Some of them are look at mutation. We then combine these to create a consensus set. Just to give you an idea of how the mutational burden somatic variation calling works. The observation is that when you move from one end of the genome to the other, due to epigenetics, regions of open chromatin. And the primary reason for this is that open chromatin is more accessible to the of modeling background mutation rate is you take all the epigenetic histone marks, as well as things like transcription rate, early replication, which you get from a reference set such as ENCODE and the epigenome roadmap, and you use that to train models of the mutation rate. This is just to show that there's very strong internal correlation between one epigenetic mark and the other, and it correlates very strongly with the mutation rate shown here in the right panel. You then, for each tumor type, you build up a model of the background mutation rate. Here we're looking at liver hepatocellular carcinoma. We have the epigenetic marks, and then you look for deviations from this. So if your mutation is a hotspot for mutation UC16 falls on the curve, so we would call it a passenger. We would expect from the epigenetic marks, we call that a driver mutation. This, by the way, was the work, the driver power algorithm that was developed by my graduate student, Shimin Shuai. This consensus driver calling and filtering process is, we identified in total 705 unique driver substitutions in 179 genomic elements. Of those 179, 143 were in protein coding genes, and almost all of them are previously known driver genes from exome sequence, which is a good positive control. The remaining 100 elements, 103 unique hits in those, and they are distributed among UTRs, long non-coding RNAs, and small RNAs and microRNAs. We also, I'm not going to go into this in any detail, but we also did a similar process for structural variations. We identified 53 significantly recurrent chromosomal rearrangements. Most of them are novel. All of them, however, involving rearrangement brings a strong tissue-specific enhancer into apposition with the cancer gene, increasing its expression. So that is that's emphasizing found with targeted sequencing or exome sequencing. Now, the surprise that came out of this is that there are there's about an order of magnitude more coding drivers than non-coding drivers, at least given the power of this data set to detect them. The vast majority of the non-coding drivers are in the promoter of the telomerase gene, which has previously been identified. About half of all tumors have some sort of alteration in TERT. Then we had a series of mutations in enhancers. So here's an enhancer in P53, the promoter of PIK3, and a series of UTR mutations. Now, the thing that stands out here is that these are primarily mutations in regulatory sites for known cancer driver genes. And that's both good news and bad news. The bad news is that we didn't discover all that many novel non-coding drivers. The good news is that it does suggest that we are saturating the cancer genome and have discovered most of the key pathways and genes at this point. And so we're seeing multiple new mechanisms for affecting genes that we've already seen. And when you add the coding and non-coding drivers together, you're able to explain 95% of cases. Only 5% of cases had no driver gene that we could detect at all. And this is just showing the distribution. The concentric rings indicate so each spoke is a different patient. The outer ring is our copy number alterations followed by rearrangements, non-coding mutations, coding mutations, which are very dense. We can then see that when you rank order the number of patients, that P53 is the most common, followed by CDKN2A, and so on, which is, and for each one, you can have either a coding mutation or you can have a non-coding mutation. You add them up together, it becomes an impressive proportion of patients. Well, the other interesting thing about this is, on average, for all patients, there are 4.6 drivers per tumor, which is remarkably consistent from one tumor type to another. So I'm going to tell you a quick but fun story about what you can do when you have a big data set in the cloud. And this is a novel non-coding driver mutation that we found in collaboration with Mike Taylor. So while the Pan-Cancer project was in the final stages of publication, I got a hot tip from my collaborator, Mike Taylor at Sick Kids in Toronto, who's doing whole genome sequencing on sonic hedgehog subtype of pediatric medulloblastoma. He found that 50% of these medulloblastomas seem to have a mutation hotspot in the small nuclear RNA that codes for the U1, or the gene that encodes for the U1 small nuclear RNA. This is the key portion of the spliceosome. This was highly unexpected because this is a evolutionarily conserved gene that goes way back to the root of the eukaryotes. However, he came to me with medulloblastoma cases and asked if we could pull something out of pan cancer to see if it was in other tumor types. So I went to a graduate student, Shimin Shuai, who had done the work on driver power, and asked him to search the pan cancer data set for the presence of mutations in U1. Now, ordinarily, this would have been a very difficult task because the U1 gene is just 150 base pairs. And if you wanted to interrogate that across 2,000 odd tumors, you would have to download 800 terabytes of data just in order to find 150 base pairs from each one. However, because it was in the cloud, he could just write a script against it and use slicing to interrogate this 150 base pair region. And it took him just about an afternoon to write the script and to do the mutation calling. And in fact, he found this kind of a very striking hotspot at position 3 of the U1 gene. There are actually 2 of them. One is an A to G, which was present in about 19% of medulloblastomas and 1 pancreatic cancer. This is the one that Mike Taylor had found. And then there was an additional A to C mutation that is found in significant numbers of chronic lymphocytic leukemia, B-cell lymphoma, and hepatocellular carcinoma, as shown in the bar chart over here. So the question was whether this mutation is changing splicing. And in fact, and you may not see it because it's cut off, but position 3 of the U1 snRNA binds to position 6 of the 5' splice site and is part of the recognition complex for the 5' splice donor. And so the hypothesis was that when you change an A at position 3 to a G, or change it to a C, it's changing the recognition site for the spliceosome. And in fact, that's what we found using the transcriptome data, which had been very conveniently gathered by the PCAWG project. In CLL, we found 3,000 differentially spliced introns and over 1,500 differentially spliced genes, about a quarter as many in hepatocellular carcinoma. And in fact, the pattern that we observed is exactly what we would have expected. In the, we expect that the A to C is going to cause U containing introns to be downspliced. There'll be fewer of them, and ones containing cryptic sites containing a G at this position would be upspliced. And that, in fact, is what we saw. So we then experimentally validated this in collaboration with Mike Taylor and Elias Campo and Jose Puento at University of Barcelona and Ovidio, and showed both in patient cells and in tissue culture cells that we'd engineered that we could reproduce this, this pattern. And we also showed that multiple known cancer drivers are differentially spliced in patients that have the U1 mutation. So are there clinical applications for this? Well, yes, there is. So first of all, it turns out that in CLL, as well as in hepatocellular carcinoma, the presence of this mutation is a bad prognostic biomarker. We're showing that the time to treatment in CLL, so when the patient becomes symptomatic and needs to be treated, is greatly accelerated as shown in the red curve. These are patients containing the U1 mutation versus those that do not contain the mutation. And the time to treatment is roughly similar as CLL patients who have a mutation in the SF3B1 spliceosome protein, suggesting that there's a common mechanism of splice deregulation there. The other thing that you can do here is to develop a neoantigen style cancer vaccine. Because of the altered splicing, we expect these genes to have to create a bunch of neopeptides, and they're going to be they're similar, if not identical, neopeptides from patient to patient, because it's a very deterministic splicing alteration. So we're now working on cancer vaccines based on using proteomics to identify these novel antigens. Finally, we've just started planning out a project in which we're engineering a U1 mutation specific cytolytic virus by taking a herpes virus and replacing the early genes that have normal splice sites with ones that have the U1 mutation altered splice recognition sequence. And the idea here is that when the virus then is given therapeutically to a patient, only cells that are expressing the U1 mutation are going to lead to active virus, which will then undergo replication and ultimately lyse a cell, whereas the wild type cells will not support viral propagation. And this is in collaboration with Dr. John Bell at the University of Ottawa. Okay, so I have a few more minutes. In the last few minutes, I'm going to talk about things that you can do with the passenger mutations rather than the driver mutations. So let me refresh your memory that the epigenetic state of the cell of origin predicts the mutation rate in the cancer that's derived from it. So as you all know, every cell type has its own distinct chromatin profile representing its state and its gene expression pattern. Because the cell state correlates with the cell identity correlates with epigenetic state, and epigenetic state correlates with the mutation rate, you can actually superimpose the mutation rate onto the open-closed chromatin pattern, and they almost exactly mirror each other. And this is work from Gadi Getz and colleagues from about 2 years ago. When we had the PCAWG set, we could ask the question, can we use the mutation rate, the distribution of passenger mutations, to identify the cell of origin of the tumor? And the idea here, the clinical application of this, is that there's a clinical problem called the tumor of uncertain primary, in which a patient comes in with a metastasis, but the primary has regressed, and attempts to identify the primary radiologically fail, and so you don't know what the tumor type is. Pathologist has to guess by looking at the histology or using a series of special stains to try to identify cell-specific markers. Our hope was that we could supplement the pathologist with a machine learning system which takes the genome sequence, applies a machine learning, a neural network, in order to identify the cell of origin, the tumor type, from its pattern of passenger mutations. So this is work done, I'm going to skip that. This is work done by my graduate student, Wei Zhao, my collaborator, Quaid Morris, and his graduate student, Gurneet Atwal. And so what we did is we took two distinct features of passenger mutations. First, we took mutational signatures, what the allele was. A is replaced by G's, C is replaced by T's, and so on, which is a signature of the mutation process, the etiology. So smokers, for example, have a very distinctive pattern of single nucleotide variations. And we also took the mutation distribution across the chromosome, which indicates the tissue of origin via this epigenetic matching. Trained a convolutional neural network with a training set taken from PCAWG, tested it with a held out set and independent sets, and then evaluated the accuracy. And it actually works quite well. When we run it on the PCAWG, a held out portion of the PCAWG data set, we're showing here, I'm showing you here a heat map of the predicted, the true labels versus the predicted labels. We show that we have overall a 91% overall accuracy, with accuracies ranging from 99% for certain tumors down to about 60% for stomach, which is frequently confused with esophageal cancer. We also validated it on independent sets. We went to the literature and found another 1460 primary tumors with whole genome sequencing. It had 88% accuracy in that. And we also took metastatic tumors from the Harvard Medical Foundation, found 83% overall accuracy. Remember, these whole genomes were not called with exactly the same pipeline as PCAWG, and so we don't expect it to do as well. So given that, it's fairly accurate. So I'm out of time here. I'm just going to go to the summary. So we've created a lasting community resource for understanding cancer biology. Coding mutations are the majority of cancer driver mutations, but non-coding mutations and structural variations play a contributing role for the same pathways. We can explain all but 5% of tumors by combining coding and non-coding events. And although the non-coding mutations are infrequent, it's still worth looking for them. For example, the U1 snRNA gene mutation may be particularly fruitful. And finally, we can distinguish the tumor type and cell of origin by looking at the distribution of passing mutations. And I'll just put up the acknowledgement slides here and take questions if we have time.
Okay, thank you very much, Lincoln, for your talk. We will have only little time probably for questions. Please go ahead, Antonio. There is a question, I think, from Anne.
There's a question from the chat from Jana. So she said that 43 million SNVs sounds like a large subset of all possible mutations. Do these usually affect the same regions as germline SNVs? Is there some obvious differences in locations that distinguish drivers? Are drivers more like germline SNVs in terms of locations?
Okay, well, it's a whole bunch of questions there. So the 43, this is the 43 million SNVs are distributed differently from one tumor type to the next. Most of them, the vast majority of them, are passenger mutations. Most of them are subclonal, so they're arising later on in tumor evolution and are present only in a proportion of the cells. And there are many, many more mutations that we did not have sufficient coverage to discover. So we were powered to discover mutations present in 10% or more of the tumors, the cells in the tumor, but there are many more. If you go down to the 1% level, you'll probably increase that number by a factor of 5 or 10. And then I don't remember what the other questions were. Other parts of the questions were. Is there a difference between germline calling and somatic calling? Yes, in fact, it's much more difficult to call the somatic variants because of the presence of subclonal mutations. We're not looking for something that's at a 50% allele frequency or 100% allele frequency, as you would in a germline.
All right. So then two more questions from Jill. How do you ensemble the colors for higher prediction? Did you use all colors and ensemble them? And also, can we say clonal mutations are most likely driver mutations in cancer?
Yeah, both are really, really interesting questions. For practical reasons, we didn't choose all the callers. we chose the subset that we felt we could run in in a reasonable length of time. So we chose 3 for substitutions and 4 for indels, and that was based on both on the performance and benchmarking, as well as how well they worked when we created the ensemble, because some of them are more similar to the others and they don't contribute additional information. Then the other question is, there are both early drivers that tend to be clonal and late drivers that tend to be subclonal. And the ones that, there's a very limited number of genes, roughly 10, which are early drivers that we tend to see across multiple tumor types. It's a core of things which initiate cancer. After that, there appears to be a period of differentiation and adaptation. Well, I shouldn't say differentiation, but adaptation and selection, probably in response to immune system, in response to therapy. Some patients did receive therapy before their tumors were surgically removed. And we see another 60 or so common late mutations, and some of them are very interesting, such as mutations in HLA and beta-2 microglobulin, which are clearly the result of immune selection. So there's a whole paper, there's a great paper in Nature called The Evolutionary History of 2,600 Tumors that goes through this in great detail. It's really fascinating reading. In fact, for each tumor, we were able to, or that group, I wasn't part of that group was able to identify the evolutionary history of which mutation came first. And the cool thing about this is some initiating mutations occurred a decade to 5 decades before diagnosis. So showing that there's a big window where we could potentially detect and treat tumors.